I'd like to start by introducing Dr. Michael Lane. He came from the land down under, like myself. He finished his undergraduate studies at the University of Tasmania, which I strongly recommend. If you haven't visited it, it's a great place to go, so make sure you put it on your travel list. He did his Ph.D. with Dr. Norman Sanders at the University of Melbourne, looking at cellular and molecular responses to injury in the developing spinal cord of the opossum. After postdoctoral fellowship training at the University of Melbourne, Dr. Lane came to the States to undertake postdoctoral work with Dr. Paul Ryer at Florida State University, and he continued at UF in a faculty position in 2009. Here, Dr. Lane became interested in respiratory function after spinal cord injury and the spinal circuitry involved in respiratory activity. Dr. Lane is currently an associate professor with the Spinal Cord Research Center at Drexel University, where he's been since 2013. He currently has significant research funding through an NIH R01, as well as awards from the Wings of Life, Craig H. Nielsen, and Mosley Foundations. Dr. Lane is also the co-director of the T32 Spinal Cord Injury Training Program at Drexel, and he's the current president of the American Society for Neural Therapy and Repair, which is a plug. It has abstracts and travel awards that are due this month, and it's a great meeting, so I strongly recommend it as Mike's PR person. So Dr. Lane's recent work has continued to focus on plasticity in the respiratory system after high cervical spinal cord injury, as well as interventions to improve respiratory outcome in this paradigm, including epidural stimulation and the transplantation of neural progenitors and V2A interneurons, the later work of which he'll present more of today. So without further ado, I'd like to introduce Dr. Michael Lane, who will present his work in today's seminar entitled Neural Cell Transplantation to Treat the Injured Cervical Spinal Cord. A warm welcome, Mike.
Thank you very much, Damien. Thank you very much for a fantastic introduction. And thanks again to everyone for having me speak here. It's a pleasure to have the opportunity to virtually speak and hopefully one day come down and visit again. Today I'm going to be talking about probably the main focus of my research team's work here at Drexel, which is looking at transplanting neural precursor cells to essentially promote plasticity and repair of the injured cervical spinal cord with a focus on respiratory networks. I want to start by acknowledging many of the people that have contributed to this work. Firstly, many of my collaborators in the Marion Murray Spinal Cord Research Center. Listed here are people who've collaborated primarily with the work I'll be touching on today. I have many external collaborators around the US and overseas as well that have contributed to a number of different projects, primarily based on cellular therapies and enhancing plasticity. And I want to, of course, thank you the financial support from the NIH and the foundation to support our ongoing research. This is my research team, pretty much most people on my research team, which comprises graduate students, medical students, undergrads who contribute to this work. And I'll touch on the individuals who have contributed to each different piece of data as we go through some of the slides. And I also want to thank our SEI consultant, Jake Chaufin. He's an individual living with cervical level spinal cord injury, meets with us on a regular basis to talk about our ongoing work and how his experiences can offer us some more input as to the way we approach our research, teaches us a little more about people living with injury, and gives him a chance to learn about what it's like to do research at the bench. So as Damien said, the focus of our work is on cervical level injury, plasticity, and primarily respiration. So this audience obviously knows the majority of injuries occur at cervical levels. That number, the proportion, is actually going up each year. And at countries outside the US, it's even higher than that. It's higher than 60%. So in Australia, I think it's about 75, and in Japan, even higher still. So the number of people with deficits of quadriplegia and a range of other motor and sensory deficits is increasing with that proportion. Among the many deficits that arise after cervical level injury is impaired breathing, which is obviously life-threatening. It's a key contributor to morbidity and mortality. More often than not, individuals with these injuries require assisted ventilation because they cannot breathe by themselves. And even those that can, which is essentially the population we are modeling preclinically, those that can control their breathing still have deficits that place them at risk of going into respiratory arrest later in life, or even deficits that often go unnoticed, like sleep disordered breathing. So here we're looking at the cervical spinal cord of adult human and the adult rat. These are actually images that I drew in Illustrator based on the actual anatomy of the cervical spinal cord from these two animal models, not to scale, obviously. But from these, this cervical spinal cord, we can start to look at underlying networks. And this is essentially what we do. We study the anatomy of the injury. We look at the physiological output, so functional output from specific networks within this injured spinal cord. And we can study how neuropathological deficits may or may not contribute to plasticity, and how that plasticity may lead to some improvements in our underlying networks that we study. So cervical level injury can occur, as you know, in a vast number of different ways. Essentially in people, it's a mix of compression, contusion, laceration, and completely different in every individual. In our preclinical animal models, the way we model cervical injury is in these three examples shown here. The first is C2 hemisection, which I won't be speaking a lot about today, is a model that's developed about 130 years ago now, so many, many years ago, to study essentially respiratory plasticity after injury. So it's a very well used example of plasticity within the injured cervical cord. We are using contusive injuries, primarily the middle one here, the lateralized C3-4 contusion, which is the level at which most injuries occur in the patient population. And it results in neuropathological consequences which relatively closely resemble those seen in the injured human spinal cord. Okay, so how do we go about studying the networks of respiration? We focus on one, the phrenic network, which is comprised entirely of neurons, motor neurons and interneurons in the cervical spinal cord, innervated by a descending input from the brainstem. Because it's located entirely in the cervical spinal cord, obviously cervical level injury compromises this network. But it also provides us with a system that we can study very, very effectively with anatomical traces and a range of battery different functional outcome measures. So firstly, we can use retrograde traces to back label from the diaphragm, which is what the phrenic network innervates, two phrenic motor neurons distributed throughout the C3 to C6 levels of the spinal cord. You can see a distribution of motor neurons here. And this is an actual example of a horizontal section through the adult rat spinal cord, showing you the distribution of labeled phrenic motor neurons. We can also start to look at descending drive by using anterograde traces. If we inject the ventral respiratory column in the medulla, we can map the distribution of that descending drive onto our phrenic network, which for a large part directly innervate via monosynaptic connections onto our phrenic motor pool. So here you're looking at an image of the phrenic motor nucleus in green, phrenic motor neurons, and they're being surrounded by these axons and boutons from descending axons in the ventral respiratory column, shown here in red. Using a transneuronal tracer, pseudorabies virus, we can also start to map out the distribution of spinal interneurons. And these are a population of extremely heterogeneous cells that can regulate motor output. And these cells are key to what we study in basically all of my research. We think that these spinal interneurons are integral elements of plasticity. They can change what they do spontaneously after an injury, and they can be harnessed therapeutically. So we started by mapping their distribution quite some time ago now to show that these interneurons firstly exist in the spinal cord. They're premotor interneurons connected to phrenic motor neurons, and they're distributed throughout a range of different regions, from dorsal horn to intermediate grade to ventral horn. Some of our research is also looking at primary afferent input, so the afferent feedback that comes back in at these cervical spinal cord levels and feeds onto this spinal phrenic network and regulates that motor output. And we can do that by using, again, transganglionic traces that pass through the dorsal root ganglion, as shown here, and label these primary afferent fibers coming into the spinal cord. Lastly, aside from the direct input from the ventral respiratory column, we can use immunohistochemical markers to start looking at neuromodulatory pathways. Here we're showing serotonin because it's known to be involved with modulation of many motor and sensory functions, including respiration, but it's also a key component of plasticity in regulating plasticity after spinal cord injury. So an example of some of the immunohistochemistry we've been doing to start mapping the distribution of axons onto our phrenic network. Then we can start looking at In addition to this large battery of anatomical outcome measures, some of the measures we can use for assessing function. And we start by looking at breathing behavior. So a behavioral outcome measure of respiration is essentially ventilation. So measures of ventilation would include breathing frequency, tidal volume. We can do this with a plethysmograph, measuring these basic physiological parameters. But it doesn't tell us specifically what individual muscles within the respiratory networks are doing. To do that, we have to go to electrophysiology. And so we can actually record directly then from either the phrenic nerve innervating the diaphragm or the muscle itself and get diaphragm EMG. When we combine these two different functional assessments, we can start looking at diaphragm contraction on the side of the injury, so ipsilateral or contralateral injury, the other side, which is usually spared for the most part by these lateralized injuries. And we can see how change in muscle activity contributes to each breath. So you can see how muscle contraction directly contributes to our behavioral output. So what happens after an injury? This is a similar schematic now showing you the injured phrenic network with an injury right in the heart of this phrenic motor pool. So in green, we have our spinal phrenic network, the motor neurons innervated by our interneurons. In blue, we have descending input from the medulla, which has been compromised by our injury. So we have denervated networks below the level of injury, as well as a massive loss of tissue and cells at the site of the injury. When we use our diaphragm EMG to record activity on the side of the injury, you can see the output, the height of the output or the amplitude is decreased. So what you're looking at here, for those that aren't familiar with looking at a physiological trace, this is actually a raw output. I'm not sure, hopefully you can see my cursor. This is a raw output of the diaphragm contraction, which is each inspiratory event. So that when the animal breathes in, the diaphragm contracts. And this is essentially expiration, where it's relaxing. On the spared side, contralateral to injury, the muscle is contracting far more effectively. So this is attenuated output on the side of the injury. Essentially means the animal can breathe by themselves. They're not ventilator dependent, which we cannot do, ethically speaking, in these preclinical models. We can study this deficit, however, and it's a twofold deficit. So the diaphragm firstly shows signs of paresis, but it also shows respiratory insufficiency, something seen clinically. And that is where the diaphragm is unable to work harder under increased conditions of respiratory drive, either by lowering oxygen or increasing carbon dioxide. We can drive the respiratory system harder and make it respond to this increased drive, where normally it almost doubles output. On the side of the injury, it's incapable of doing that, which means if an individual or an animal is compromised and the muscle cannot appropriately respond or compensate, they may go into respiratory rest at that moment. So the focus, as I said, of our research is on these spinal interneurons, these neurons that innervate motor neurons, modulate their function, and essentially, we believe, are key elements of ongoing plasticity. As many of you in the audience will know, that spinal interneurons have been shown in a vast range of different motor and sensory systems to contribute to plasticity after spinal cord injury, because they have a remarkable capacity to reorganize their connections and change their function. And they adapt to changing conditions quite easily. This amazingly plastic ability makes them very useful to contribute to spontaneous improvements after a spinal cord injury. And that alter their connectivity to do so. But it's a very heterogeneous population of cells. As this example here of a cross-section of the rodent cervical spinal cord will show you, there are a huge number of thousands of neurons at any given section of a spinal cord. And shown here in red are all the neurons in this one particular section. In blue, we're standing for acetylcholine. So it's a number of cholinergic neurons. And already you can start to see, in addition to motor neurons down the bottom here, we have a sprinkling of cholinergic interneurons, which are one of the populations we've become interested in. But it also starts to speak to the massive heterogeneity of this population of cells. There's a number of different cells doing a number of different things, and they keep changing under different conditions. So that plasticity is wonderful therapeutically, but it also makes it difficult to study. So we believe, as I said, interneurons are key to plasticity. The question, of course, is, given this heterogeneity, which interneurons contribute beneficially to plasticity, and which, if any, might be contributing to maladaptive plasticity, which is something that the field has begun to study more recently. We know that both types of plasticity exist, and so we are interested in trying to map out those two issues. In order to better understand the types of interneurons that exist within the spinal cord, we go back to development. And in the last couple of decades, developmental neurobiology has come ahead very quickly in identifying populations of spinal interneurons that have been, for the most part, classified as cardinal classes, which are progenitor cells throughout the developing neural tube, as shown here, which can start to identify different subsets of interneurons. And this is highlighting here in red, CHUX10, which is just one example of a transcription factor identifying one subpopulation of interneurons, the V2A excitatory premotor interneurons. As we start to look at these different populations of cardinal classes, you can see down the side here, there are dorsal and ventral populations, very crudely initially broken up into sensory and motor precursor cells, which is not always entirely the case. We focus primarily on the ventral populations because they have been associated with motor functions, and also because prior work in our hands has shown that ventral populations may be beneficial for plasticity within respiratory networks. So we've begun to start to study firstly V0s, which comprise these cholinergic interneurons I mentioned a few moments ago, V1 interneurons, which are inhibitory, so they actually attenuate motor output, and these V2A excitatory cells, which show excitatory inputs to respiratory motor neurons and can enhance activity. As we map the distribution of these cells over time, we can get a feel for where these different populations of neurons are moving to and how they connect to different networks. In our hands, of course, the respiratory network. So of the two populations I'm going to tell you about today, our focus is primarily on these V2A excitatory ipsilaterally projecting spinal interneurons and also the V0 cholinergic cells. A few years ago, I had a graduate student working with me, Lana Joludiva, who's actually I think wrapping up her postdoc now and looking for faculty positions. She actually spearheaded this work looking at V2A interneurons in the naive spinal cord and mapped out their projections within the Phrenic network to firstly identify that they were even there. Using a transgenic line where GFP was expressed within CHUX10 positive cells, we could map the distribution of these V2A excitatory interneurons within the Phrenic network, which is labeled with our pseudorabies virus. And then quantifying these in the naive and injured spinal cord, we actually saw a significant increase spontaneously in the number of these cells that were connected, although with some heterogeneity between animals, which is quite an interesting point. And I can come back to that later on if we have time. But it does show even in a spontaneous condition, you see this anatomical plasticity, which coincides with the recovery of diaphragm output. The cholinergic spinal interneurons we can also study using immunohistochemical markers as well as transgenic lines, which we have in-house. And we can map the distribution of cholinergic cells within the Phrenic network in pretty much the same way. And when we do that and look at how they change their connectivity after injury, again, we see that there is an increase in their connectivity of these cholinergic interneurons to the Phrenic network post-injury, but it's a fairly modest increase compared to what we've seen with our V2A cells. But we thought this was interesting enough to pursue further. And so we began to look at who's studying cholinergic interneurons and who can help us learn a little bit more about them. It's spearheaded a collaboration with Gareth Miles at St Andrews University in Scotland, who's been studying these populations of cells and their contribution to motor function plasticity within the intact spinal cord and locomotor function, and also following ALS. His early work actually began in respiration. So when I spoke to him about this, he was keen to help us start identifying these cells and how they might contribute to breathing normally, which is the case for his lab, and post-injury, which is what my research team brings to the table. So initially, Margot Randelman, a graduate student in my lab, and a postdoc working with him, Simon Sharples, started this project immediately prior to COVID, which shut this down very quickly. And unfortunately, we've made slow progress since 2020 because of that. Partly due to the fact we lost the transgenic lines that we had access to and Gareth had access to. So we've started to remake those from scratch, and we have those up and running now. And so now we have a new graduate student working with Gareth and myself that's spearheading this project to start studying the role of these interneurons. Hopefully in the coming year, we'll know more about what they can do spontaneous for plasticity after cervical level injury. So we do know, however, based on this initial work, that cholinergic V0Cs and our V2A spinal interneurons exist within the uninjured Phrenic network. We know from Lana's work that the V2As spontaneously contribute to anatomical plasticity after injury. And we know from recent work by Margot Randelman that the cholinergic spinal interneurons increase their connectivity even more so after injury and training. And I'm going to touch on that in just a second. With that in mind, of course, the question is, Now we identify sums of these spinal interneurons. How can we therapeutically harness the potential of these cells and better use these neurons and their connectivity to improve outcome? So the two ways my research team look at therapeutically enhancing plasticity to firstly harness the potential that we all know exists within rehabilitation or activity-based therapies or even neurostimulation. And the second, which is the larger part of my ongoing research, is looking at strategies for repair, and that's using the cell therapy. Firstly, I'm going to touch on rehabilitation just briefly because this directly relates to our ongoing cell therapy work. So the two ways we look at enhancing or driving neural activity within the spinal cord can be broken up into neural interfacing and activity-based therapies, as outlined in this diagram here. And these are all sorts of approaches that we've used primarily through collaboration with individuals over the past 10 years. The one I'm going to touch on just briefly is an activity-based therapy, respiratory training, where we essentially change the inhaled gases that an animal might breathe in order to stimulate breathing, and we can do so intermittently. We know that by doing so, we can drive plasticity both supraspinally and spinally, and even throughout the entire neural axis, just by using this intermittent hypoxia or intermittent hypercapnia training regime. The focus of this work still is on these spinal interneurons, and that is, can with these sorts of training strategies enhance the connectivity of beneficially connected spinal interneurons? So the way we go about treating these animals after an injury is primarily with intermittent hypercapnia. This is a treatment that we've been pioneering in our lab, which builds primarily on the work done by Gordon Mitchell originally at the University of Wisconsin, now at the University of Florida. And intermittent hypoxia has been used preclinically in a range of intact and injured spinal cords, and even in ALS, to enhance respiratory plasticity and even non-respiratory plasticity in locomotor systems. And because of its benefit shown across multiple different systems, it's also been translated to the clinic in some initial clinical trials around the country. So building off the heels of that, and the strategies based around this intermittent exposure or intermittent changes in activity, we thought intermittent hypercapnia might have a similar effect. And so it essentially consists of 5-minute bouts of either increasing CO2 or lowering oxygen in the case of hypoxia, with a 5-minute rest in between. So it's sort of like giving your diaphragm this intermittent training, which is like doing sets at the gym, where they work out and they have a rest, they work out and have a rest. And if this is done repeatedly over the course of a day and then done daily, 5 days a week for 4 weeks, that sort of an intense training regime actually promotes plasticity throughout the neural axis. We then start to study changes in the network using those same outcome measures I explained previously. So we can map descending drive to our injured phrenic network, back label our motor and interneurons, and start to record the diaphragm output following injury and treatment. And what we've shown is that with immunohistochemistry, looking at distribution of serotonin, and we compare both intermittent hypoxia and intermittent hypercapnia, this training for 4 weeks can actually significantly enhance the degree of serotonergic input onto our phrenic network. Significant with some variability with intermittent hypoxia, but even greater degree of serotonergic growth and sprouting onto our phrenic network with intermittent hypercapnia. When we look at the connectivity of our interneurons with a focus on cholinergic cells in this case, we can see that the number of cholinergic interneurons connected to our phrenic network also is significantly enhanced with just 4 weeks of intermittent hypercapnia training. And this is again looking at diaphragm output, showing you that with the yellow trace, we're also enhancing diaphragm amplitude during each contraction. The average diaphragm output is significantly improved both under eupnea conditions, and we improve their response to a respiratory challenge. When these sorts of conditions, if they could be translated through into the patient population, would be life-altering. So in summary so far, spontaneous changes do occur within this injured network. We know that there's spinal plasticity within multiple systems, where here we're studying it in the respiratory system. We've identified changes in spinal interneurons and how they're mediating this plasticity after cervical level injury. We can map this connectivity after activity-based therapies and show that it's enhanced. So rehabilitation does enhance the connectivity of spinal interneurons within this respiratory network. Of course, we're interested in seeing how we can better characterize this training with different forms of respiratory rehabilitation in future ongoing studies. But to feed into this other aspect of our ongoing interests in promoting plasticity, we're interested in again, targeting these interneurons and understanding specifically how we can use interneurons directly to enhance plasticity. And that focuses on these, again, V2A cells and cholinergic interneurons, with a focus on now not rehabilitation, but repair. So rehabilitation and activity-based therapies and neural stimulation are fantastic because we know that they, across multiple different systems and preclinical animal models, have shown efficacy, but there's always going to be a limit because there's still an injured underlying network. There's a part of the network that's missing. And so the limitation is going to be based on the fact that these essentially harness activity in what's left, what's spared by the injury. And so in order to enhance plasticity further, we're interested in trying to promote a degree of additional repair via cell therapy. So this now builds on decades worth of work looking at neural precursor cells transplanted into the injured spinal cord. And if we transplant these cells into the injured spinal cord, we can map their distribution. Primarily, people are now using fluorescently tagged cells so we can see where they've been placed and how they connect and what they become. Work on our hands, like many others, have shown that transplantation of neural precursor cells, so the developing neurons and glia of the spinal tissues, can actually survive this inhibitory environment of the internal milieu of the injured spinal cord and develop into neurons and glia, providing us with essentially the building blocks to repair the injured spinal cord. Not only that, but like many others have shown, Paul Ryer, Itzhak Fischer, Mark Tuszynski, and many more, these same populations of cells extend their neurites out into the intact regions of the spinal cord. They have ascending and descending neurites, which may connect with the surrounding tissues. Specifically of our interest is the fact that these cells can integrate with our injured phrenic network below the level of injury and start to innovate the motor neurons and interneurons within the phrenic motor network. And so what you're looking at in these photographs on the side here are essentially NPCs or neurites for NPCs, shown here in green, wrapping around the phrenic network, which is shown here in red. So in this first picture up the top, we're looking at a spinal interneuron, which is surrounded by boutons and axons from our donor cells. Similarly, when we look at the phrenic motor pool, so our motor neurons, below the level of injury, those same motor neurons are receiving multiple inputs from these donor cells tagged with GFP. Using our pseudorabies virus, we can also backlabel these cells and show that donor neurons that survive in this transplanted tissue readily connect to our phrenic network and are also labeled with pseudorabies virus. So in order for these cells to pick up pseudorabies virus, they must be synaptically integrated to that donor network. We're keeping in mind this tracer was put at the diaphragm. So essentially these donor neurons are connected to the network controlling that diaphragm where we placed our tracer. That image also shows that with immunohistochemistry, a subpopulation of these connected cells are also cholinergic, which are now our subset of interest, our V0C cholinergic interneurons. Using immunohistochemistry, we can start to then also look at inputs to our donor cells from supraspinal regions, so either by tracing from our respiratory centers in the brainstem, or by looking at serotonergic input, we can start to map the distribution of input to our transplanted neurons. And again, we see that there's input both from axons within the VRC, although to extremely limited extent. And with serotonin, we do get a little more ingrowth of these modulatory fibers, which seem to grow more readily through otherwise inhibitory environments, and they certainly do connect with our donor cells. So essentially, we have the capacity to create an anatomical relay with these donor cells. And that's something that many people have shown now using a range of different techniques. Here, we're demonstrating it within the phrenic network. We've also become quite interested in the primary afferent input, which John Halley and Paul Ryer a couple of decades ago now showed that these cells readily, spontaneously grow into donor cells. We're interested in seeing how they might also be regulating that motor output and potentially contributing to plasticity in non-respiratory networks. So we, like many others, have shown quite comprehensively by transplanting neural precursor cells, essentially derived from a primary source of tissue from developing spinal cord. So they can either be fetal tissue itself, or it can be cultured neural precursor cells, as Tuszynski and others have shown. These cells survive, provide us with new populations of neurons and glia, and they integrate very effectively with the surrounding tissue. However, two main issues my research teams become very interested in is the degree of survival and integration is incomplete and inconsistent. And to me, that's one of the issues that we need to address before thinking about how this therapy might be most beneficial therapeutically and translationally. Any level of that variability is going to feed into variability among the patient population, which we want to avoid. A second issue which we're trying to address is that the phenotype of these cells was never really comprehensively characterized. So can we better understand who these cells are? We know they're a heterogeneous mix of precursors. Can we start to refine that down into populations that we know might be beneficial? So in order to start addressing this, we firstly started to look at donor cell phenotype. And specifically, we became interested in, given that these are spinal-rich precursor cells, which spinal interneurons, or maybe even motor neurons, are present within this donor cell tissue. And so we ran qPCR to start studying within cells that were either prepared in culture or straight from the dissected out of the spinal cord directly, what sorts of precursors exist. When we look at this and looking at a range of different transcription factors, which are listed along the bottom here, which correspond to these cardinal classes of spinal interneurons, we can see how in white, we have our normal developing spinal cord. By dissecting this tissue out and culturing it in preparation for transplantation, we are changing the population of cells we have available for transplantation quite significantly. Firstly, our V0 cells, which comprise or contain our cholinergic interneurons, and our V2A excitatory interneurons are significantly reduced. The expression of those transcription factors is ramped down a lot. So we are potentially losing two populations of interneurons that would be key to repairing this injured Phrenic network. We've also lost motor neurons, which is not something that is novel, that people have known for quite a while, that we lose motor neurons when preparing this tissue. But the loss of these specific interneurons could be detrimental, especially considering one that we seem to retain, relatively speaking, are populations of premotor inhibitory cells, which may in fact limit the degree of plasticity we see. So we're looking at a series of different experiments over the past couple of decades and compiling some of the data. What my research team found is that if we treat the animal with a vehicle, so we provide no cells, we see very little diaphragm recovery. This is what can be attributed to essentially plasticity, but output is limited. If we transplant NPCs, so this is fetal tissue that's been cultured to provide us with a population of neural precursor cells, we can enhance that output, although it's still not complete, given that the diaphragm output of an intact animal is around 0.4. So we're looking at all the way up the top here, and it does not achieve a full recovery of our output with this NPC. But the NPCs that we've been using, this heterogeneous mix, potentially have lost the interneurons of interest, which began in a collaboration between Lana Jolo-Diva and Shelley Sakayama-Elbert, a cellular engineer at the University of Texas, to start looking at how we can use these engineering techniques to have a stem cell-based therapy to create populations of spinal interneurons that we combine with our NPCs and enrich for these V2A populations of cells and enhance output. When we did that, we found that not only do V2A cells survive and integrate with our injured Phrenic network, but they also significantly improve output. So this is a great result. It's still not a complete recovery, but we've only really optimized for one type of interneuron. The other issue here that I've been interested in is still we get this variability. So with this refined population of NPCs, albeit we've lost our population of cells of interest, we get quite a clustering of outcomes and a fairly consistent result with NPC transplants. When we re-enrich for V2As, we start to get some heterogeneity and variability again. We believe this is for two reasons. Firstly, we've re-enriched for one cell type only, and we've guessed at the dose that we need to remix them, so we need to test dosing. And we also need to look at the degree of connectivity. Are these cells connecting? And if they do so, are they connecting consistently? This led to an ongoing study to continue this work with a new student in my lab, Tara Fortino, who's now begun to transplant these same cells, but now the V2A interneurons have been modified to express the inhibitory dread receptor, HM40I. So after transplantation, months after transplantation, when we look at diaphragm output, we can then deliver clozapine to essentially turn off our donor V2A cells and dampen that activity. And so what you're looking at here is activity during one of these recordings. We're looking at baseline contractions of the diaphragm before we've provided clozapine. This is a transplanted animal. When we deliver clozapine directly into the transplant, in this case, we see that there is an attenuation of output from the diaphragm. So by turning off directly our V2A cells comprised in the transplant, we reduce the diaphragm contraction. So we know those cells are there and they're connected. This shows they have been making a functional contribution to this activity. Even when we expose the animal then to a hypoxic challenge to drive that respiratory system harder, it does not show a very significant output in terms of activity. So the cells are definitely there and connected. They contribute to output. The question is now, how do we go about getting improving output so it's even greater recovery and getting maybe a more consistent recovery in each of these recipients? That's where we come back to the question of how we get consistent transplant donor neuronal connectivity with our host and vice versa. And to do that, we start to combine the activity-based therapies I spoke of earlier, these respiratory training strategies, with our transplantation. And we believe through this combined approach, we can start to get at the two major issues we've encountered, and that is refining our donor cells to the right type of cell, and now providing an activity-based therapy or a means of enhancing activity within those donor cells to the appropriate network performing the appropriate function. So here's an example of a spinal cord where we transplanted NPCs. These were not enriched with V2As. This is just a population of NPCs. We've traced these cells with pseudorabies virus back from the diaphragm, so we can count the number of donor cells that have become connected to the Phrenic network. And we can start to compare how this connectivity changes between animals that just receive cells and nothing else is done versus those that receive these cells and also undergo activity-based therapy with intermittent hypoxia training. And what we did here is compare the number of GFP-PRV cells connected to the Phrenic network under these two different conditions. And you can see that in blue, the number of cells connected is relatively small, but we can significantly increase the number of those donor cells connected to the Phrenic network, essentially on both sides of the spinal cord, by using this 4-week training with intermittent hypoxia. The question, and an additional question, of course, is when we train for this activity-based therapy, which spinal interneurons of our donor cells are connected? We know that normally a subpopulation are these cholinergic cells. The question, of course, is, does training enhance that connectivity? And preliminary data suggests that it does, that we increase number of connected cholinergic donor cells to our Phrenic network fourfold. And that fourfold increase, we're trying to now see if that correlates to improvement in functional, consistent functional improvements. So in summary, so far, we know that spinal interneurons mediate plasticity after injury. We know that spinal connectivity can be enhanced with activity-based therapies like intermittent hypoxia or intermittent hypercapnia. And we know that by transplanting spinal interneurons, we can improve outcomes. So firstly, NPCs are innately enriched with interneurons, although by preparing for transplantation, we modify that population. We know that also engineering specific subsets of interneurons to refine the donor cells we're using can more directly and specifically lead to improved outcomes when we know which interneurons we need to be transplanting. By pairing these transplantation strategies with activity-based therapies, we can start to look at also improving connectivity. And we're exploring now if there's alternative ways to enhance this even further. So to take this a step further and better understand how we can use these systems to improve connectivity and whether or not we can start to recapitulate these types of strategies with a translational approach, and that is making human spinal interneurons, I've begun a collaboration with Lana Joladiva, now that she's a postdoc at the Gladstone Institutes. She's begun working with cellular engineers to make human neurons derived from human iPS cells. And essentially what she has made from multiple different lines of human iPS cells, she's found the appropriate line to best make populations of human V2A interneurons, which can be genetically modified to also express channelrhodopsin. So we have cells that are expressing the optogenetic marker so that we can increase their activity with blue light. By creating and transplanting these cells into the same lesion in the adult rat, so it's a C3-4 contusion, just looking at the contusion site here, we can transplant these cells, and a month after transplantation, we can go in and directly record from the transplant as well as shine blue light onto the transplant to enhance output. And this is just of human V2As. This is you're looking at the recording here of diaphragm output on the site of the injury. Now you're looking at spontaneous plasticity, in this case here, with output normally before we shine blue light on the transplant. And when we place this cannula with a blue light going into the transplant and activate those donor human V2A cells, we enhance diaphragm output. So again, human cells survive, they connect to the Phrenic network, and we've confirmed this again with rabies virus tracing, and they also directly lead to improved output when we enhance their activity. So before activation, it means these cells were not entirely active, which means maybe we can enhance them further once again with activity-based strategies, enhancing or priming these cells, not only to be best connected, to be most effectively active. So considerations for ongoing work are essentially what other interneurons exist within this Phrenic network that can start to be looked at therapeutically to enhance plasticity via activity-based therapies or neural stimulation, or can even be made from transplantation. So we're starting to study different populations of spinal interneurons that may be connected to the Phrenic network, and also start to understand how they might also exist within non-respiratory networks like those contributing to upper extremity function. We've begun exploring combination therapies to promote targeted and preserved connectivities, both with our donor cells to our host and vice versa. And we're looking at using not only activity-based therapies and different types of activity-based therapies for different periods of time, so taking it even longer to see if we get even further enhancement of this connectivity and retention of that connectivity for chronic periods of time post-transplantation. But also, can we use neuromodulation, intraspinal stimulation, either directly via multi-electrode arrays that can stimulate the donor tissue, or by using optrodes that can be using light as a source to activate these donor cells to drive connectivity and activity. We're also trying to move more effectively towards treating chronic spinal cord injury. The vast majority of the work I spoke about today was treating acutely injured spinal cord, and we're trying to look at treating the chronically injured spinal cord, which is months to, in some cases, we're hoping to do a year post-injury and start transplanting these cells and see if they have similar levels of efficacy, both in terms of their reparative capacity and in terms of their functional improvement. And we're expanding our interest of our transplanted cells into non-neural cells. The focus of what I spoke about with these transplants with the neuronal elements, specifically spinal interneurons. We're also interested in the glial cells and endothelial cells. These two populations of cells that essentially do make up the cytoarchitecture of the spinal cord normally. Can we engineer these different populations to start making a tailored mixture of cells for transplantation to treat injured spinal cord? And so with that in mind, I want to thank the people that have contributed to this work. Margot Ranneman, who pioneered the intermittent hypercapnia activity-based therapy training. Tara Fortino, who's now taken over the work with V2A transplantation. Kayla Chardine, who's looking at using an array of different transgenic lines to start mapping plasticity within different spinal neuron populations. And Adam Hall, who's looking at these combination therapies, looking at transplantation combined with activity-based therapies. Alessia Nisiforo is a postdoc who joined the lab who's looking at transplantation of glial cells. Specifically, we're collaborating with other individuals at Drexel University to engineer pro-reparative glial precursor cells that can also then subsequently be reprogrammed to provide us with spinal interneurons. So it's a two-pronged strategy for transplantation and repair. Our other collaborators that primarily contributing to the work I spoke about today here at Drexel, Oscar Chung, who is an expert in human iPS cells and cellular engineering, Kimberly Doherty, whose expertise lie in the V2A spinal interneurons, Simon Gishta, who's helped us out with multi-electrode array implantation and stimulation, and Tanya Bezdugnaya, who's looking at innate plasticity within respiratory networks and sleep disorder breathing. Outside of Drexel, some of the key collaborators for this series of work were Shelley Sakayama Elbert, who's now at the University of Texas and is a cellular engineer making many of the populations of spinal interneurons I spoke about. Lana Jolodiva, who's making the human versions of these same interneurons, which we can transplant, and have also been modified to express the channelrhodopsins. Shane Lidlow, who's helping us define what is a glial phenotype that is pro-reparative and how can we best start to engineer cells to create the appropriate phenotype, not only in a dish prior to transplantation, but hopefully retain that phenotype after transplantation. And Gareth Miles is helping us to better study these cholinergic interneurons to better understand their plasticity normally and how we can study them after transplantation and repair. So the ongoing work, as I said, in terms of cell strategies is building upon these three populations of cells. The spinal interneurons, which we have been interested in on all of these projects, and we still believe are a key component to all of this ongoing plasticity and repair, but also with consideration of these two very important elements, the pro-reparative glial cells, which not only are supportive normally, but can clearly change the cytoarchitecture of the interspinal cord, promote and support repair and plasticity, and may actually serve as a target for reprogramming to provide new populations of neurons with root reprogramming techniques. And also the transplantation of endothelial cells, which themselves can provide a scaffold for repair and axon growth. Because we know from developmental studies that developing vasculature can support developing axon growth. And we also obviously know that donor tissue needs to be highly vascularized, or any sort of repaired tissue. And so essentially by using a combination of these three different types of cells, we can start looking at engineering what we hope will become a very effective cell therapy in future years. Essentially looking at different populations of spinal interneurons, pro-reparative glia, and the reparative endothelial cell progenitor cells. And with that, I'll take any questions. Thank you.
All right, thank you, Mike, for a great talk. We'll open the floor to questions if anyone has any questions. I guess I can start off with a question regarding aberrant growth. Have you looked at, I guess with so many cells being transplanted, is there aberrant plasticity or growth into areas that you would not expect that may limit your recovery?
We believe there is. We don't know for sure just yet. So one of the reasons we believe that might be the case is because, especially with our activity-based therapies, we're seeing an upregulation of a huge number of different growth factors, including NGF. So NGF may not be a good one to be promoting under some conditions, and we don't yet know how much is promoting the enhanced growth of primary afferent inputs, which could be maladaptive or aberrant, certainly making aberrant connections with our Phrenic network. What we also have been looking at is connectivity of inhibitory interneurons, the inhibitory V1 spinal interneurons, and mapping out how they connect to this Phrenic network, and maybe also limiting plasticity. And so by studying this, we're trying to best determine, is there a strategy by which we can limit that those maladaptive changes, or even prevent connectivity of cells that might be limiting plasticity? A collaborator of ours a few years ago, when we were looking at epidural stimulation, showed that if we attenuate activity in inhibitory neurons just globally throughout the cervical spinal cord, pharmacologically speaking, we significantly improve recovery, innate plasticity, and we enhance the effects of epidural stimulation. So there's definitely a network there that's connected, that's limiting the potential for better recovery.
Okay, thanks.
I had a question.
Yep.
That's a very nice talk. I'm very interested in the human V2A interneurons, and you showed that they were optogenetically activated, but the response is actually many minutes following the stimulus. So I was kind of curious, looking at other optogenetically activated circuits, that the effects are primarily established during the time of stimulation, but not so much afterward. Is there something unique about these neurons that you are dealing with that have such prolonged effects?
That's a good question, and we really don't know. We don't have a clear answer to that, I would say. I think what is happening is there is no doubt we're activating the cells immediately. I think in vitro, we've shown that. We grow the cells on multi-electrode arrays, and you can activate them and get output immediately. And you can even entrain and pattern activity in these cells in a dish. But in a transplant, we're now looking at the cells themselves to connect to a quite complex network to result in a downstream effect in a muscle. And so I think what we're seeing is essentially facilitation or potentiation of activity in a transplant that leads to a persistent increase in activity within that network, which is fascinating in itself, but we don't know how that's happening. So we're looking at trying to use combinations of both excitatory and inhibitory dreads and optogenetics to try and better tease that out. But it's, yeah, as you can imagine, it's even in this relatively simple neural network, it's a complex question to answer. Great question, thank you.
Michael, really nice talk. Thank you very much. This combination approach, it just gets more more complicated as we think about new ways to manipulate the systems. And of course, you ultimately want to translate this into people, so you just wonder what you can do and what you can't do. But the cholinergic spinal interneuron story that you gave us today, I think is very intriguing. How about pharmacological manipulation of the cholinergic neurotransmission and how that could be coupled with neuromodulation, for example, or activity-based rehabilitation?
That's a good question. I think it's certainly something that people have not looked at in this system. And maybe it's because people didn't really know that it even existed as a therapeutic target. I think for the most part, people have been very focused on the cholinergic cells being the motor neurons themselves. So the role of these cholinergic interneurons has not been well explored in most systems, let alone in the respiratory system. And I think it definitely is an approach that people could use. I think the complex issue that would be dealt with is there's not a single population of cholinergic cells. So do you begin to activate other cells that might not be these V0Cs? So whether or not that resolution is needed is an unknown at this point. It's something worth exploring. Maybe, however, while looking at transplantation, we can also start to think about the fact that these cells are crucial to regulating output. Keeping in mind that even though the V0Cs, this population of cholinergic interneurons in the intermediate gray, it's a small number of cells. So most people dismiss them because it's such a small number, but it also is the source for 90% of cholinergic inputs, these C boutons, onto motor neurons. And they are known to be cells that directly regulate the amplitude of motor output. So their functional contribution is extremely high. Their importance is key, even though it's a small population of cells. So yeah, I think you're right. We need to explore a number of different ways to target them and understand how we can tap into them therapeutically. And pharmacologically is an obvious one to begin with. We've taken a different strategy, I guess, for now.
Great, thank you. If you had strategies to induce robust axonal regeneration in descending tracts, would you hypothesize that you also have to find a way to induce plasticity in the interneuron population as well, or would one be enough?
That's a good question. I think with the strategies that I've tested in the past in the Phrenic system to promote regeneration, a really interesting thing happens. And I think it's because the interneurons are so key to ongoing plasticity. But we look at enhancing growth of axons from the ventral respiratory column, so the major drive for contributing to breathing, right? There's not a lot of growth after injury. That, much like the CST, does not grow much at all. We can enhance growth with a number of different pro-regenerative factors. But what happens rather than grow around or through the injury site is we enhance growth onto cells above the injury site. And so we're essentially enhancing connectivity onto spinal interneurons. And I think those spinal interneurons already relay that information around or even through a network with the transplant. So while I was consistently looking for growth of these supraspinal tracts, I don't think it's needed because it already defers to these polysynaptic pathways. So instead of seeing growth through around an injury site or into a transplant, I think we're getting increased connectivity of host spinal interneurons to our transplant and the transplant onto spinal interneurons. So it's never a supraspinal connectivity to the motor neuron output. I think the simplicity of those monosynaptic pathways is probably in some cases overrated. It definitely exists normally in the respiratory system, but it's definitely not needed within the respiratory system. And so I think by promoting these polysynaptic pathways, which are occurring naturally anyway, we can enhance that output. And most regenerative strategies that I've looked at seem to enhance plasticity rather than long distance axon growth.
Okay, great, thank you. Great talk.
Thank you very much.
I had another comment. So have you thought about stimulation of the respiratory centers directly? I know I would think that to try to do that in humans is risky because you're in the medulla, but there are other ways of activating these systems. For example, there's a central connection between locomotor initiation and respiratory output. We've seen this in the cat where we did fictive locomotion and we were able to increase FOS expression in the respiratory pathways. even though the animal is not actually physically moving because it's neuromuscularly paralyzed. And the locomotor centers also not only activate central respiratory pathways, but they also increase the release of serotonin in the spinal cord. So I was just wondering if maybe that's maybe a slightly safer approach, and maybe not necessarily good for locomotion with such a high cervical injury, but maybe it's enough to facilitate respiratory activity.
Yeah,
anyway,
so
just an idea.
that there are a number of different ways you can go about in stimulating plasticity throughout the neural axis, and certainly driving respiratory activity within the medulla. I looked at it some time ago, looked at trying to stimulate plasticity or stimulate even activity within respiratory centers in the brainstem. And one of the immediate complications is there's a huge amount of plasticity there normally as well. And we've only begun to touch the tip of the iceberg, but we've seen pretty significant reorganization of cells within the medulla, much like you see in the cortex related to cortical plasticity after any sort of injury. And so that reorganization happens pretty quickly after injury, and we don't know what it means. That's the first issue we need to face. The other thing is, however, we've taken many different strategies for looking at increasing activity peripherally. So we've used FES to stimulate the diaphragm. And we know that clinically this is being used around the world now, diaphragm pacing as a means for maintaining ventilation. But stimulating those primary afferents, whether it's directly by afferents through the phrenic nerve or indirectly through pulmonary receptors, right? The chemoreceptors and the baroreceptors that are involved with measuring our respiration, ventilation, they all become active when you start driving respiratory system with stimulation of the muscle. And you can get increased activity within respiratory centers in the brainstem, as well as increased plasticity centrally within the spinal cord. We've also looked at locomotor training as a non-respiratory means and how that contributes to breathing. Anecdotally, work being done in University of Kentucky with Alexander Ovechkin has shown that activity-based therapies, locomotor training and treadmill training significantly improves breathing. And I think, like you say, in many models from cat to rabbit to some canine studies, they've shown stimulation of the lower limbs directly stimulates activity of the diaphragm. We've even shown that pacing the bicep can otherwise activate a paralyzed diaphragm just by stimulating the muscle itself, the bicep. So these networks are so integrated.
What about vagal stimulation? Would that possibly?
Yeah, absolutely. And there's definitely people that have begun doing that. I've been looking at, Patrick Ganser, I know, was looking at that some time ago now, and stimulation of the vagus nerve to see if you can essentially amplify plasticity, right? And I think it amplifies plasticity within multiple different networks, including the respiratory network. Yeah, for sure. That level of integration, we believe, is a lot through spinal interneurons. We just, as you can imagine with this work, we've only really begun to identify how much these networks are integrated.
Okay, well, thank you very much for all your questions. And thanks again, Michael, for a great talk. Lots to think about, and wish you all the well with your work towards translation.
Thanks very much, everyone.